Now the last one, so this one, feature counts, will generate a list. Everybody knows how to work with a list if you have worked in R, so that's not special. But this one, the summarize overlaps function, generates a very special data type, and that's called a summarized experiment. So those of you who have experience in working with R, you know that R comes with a number of basic data types. You have vectors, you have matrices, you have data frames, and you have lists. Now for omics data, they often use more complex data types than that. A simple matrix or a simple data frame will not do the job there, because the data is so complex. And this is where the summarized experiment comes in. So originally, this data type was created for holding microarray data. But RNAseq data are very similar, so you can also use it for that. You can also use it for holding proteomics data. So every omics experiment that creates a matrix with counts or expression values can be stored in a summarized experiment. So this is one of the crucial data types for doing this kind of RNA-seq analysis. So this is why I'm going to explain. So what does it consist of? So a summarized experiment consists of a number of matrices and a number of data frames. So basically it's a combination of matrices and data frames that are linked to each other. So they are related. There are links between these different objects. So centrally to the summarized experiment, you have matrices. So you have a matrix that contains the raw counts, for instance. You will also have a matrix that contains normalized counts. When you do your analysis, you're going to normalize your counts, so the results of this will also be stored in a matrix. So you have a number of matrices here that are central to your summarized experiment. So in these matrices, the rows represent genes. So every gene is one row in such a matrix. And the columns are samples. Every sample becomes one column in such a matrix. And then linked to that matrix, you will have additional data frames. And that's these things. So you will have a data frame that contains annotation of the genes. And you can have functional annotation, so gene IDs, ensemble IDs, gene symbols, that kind of annotation. And you can also have positional annotation. So for every gene, where are the exons located? That's positional annotation. So all these annotations are stored in data frames. You also have a data frame that contains annotation of the samples. So for every sample, you have the name of the sample, you have the name of the file that contains the counts, and you have the grouping information. So which sample belongs to which group? Which sample is a control sample? Which sample is a treated sample? So that's here in the sample annotation. And then on top of that, you have metadata. So these are additional data frames that contain additional information about how you're doing the analysis. So which parameter settings are you using, for instance? That's the metadata. How can you access these data? Well, you have specific functions for that. So if you want to access the counts, you have to use the assay function on the summarized experiment. If you want to access the column, the sample annotation, you have to use the call data function on the summarized experiment. If you want to access the gene IDs, so the functional annotation of your genes, you have to use the row data function on the summarized experiment. When you want to access the positional annotation, so the annotation of the exons, for instance, you can use the row ranges function. To access the metadata, you use the metadata function. So this is what is being generated here by this function. So a matrix with counts, because that's what this function is doing, it's counting. So it generates a matrix with counts and additional data frames here that are linked to that matrix with more info on the genes and on the samples. What's the benefit of this? Why would you do it like this? Well, the benefit is that all your data are in one object. And you don't have a separate matrix for the counts and an additional matrix for the normalized counts, and then again an additional data frame for your gene names. It's all in one object, and the benefit of this is that when you make a change here in the matrix, when you remove a row here, for instance, that all the other stuff is automatically updated. So when I remove a row in the matrix that has the raw counts, then automatically that row will also be removed from the normalized counts. And that row will be removed from the gene annotation data frames. And you don't have to keep track of it yourself, which is very helpful. It would be hell if you were responsible for doing that. So it has big benefits using these more complex data types.